This is our presentation. I'm Li Zhengda, EGE 2409015. So our topic is comparison and HPLC quantification of antioxidant profiling of ginger rhizome, leaves, and flower extracts. So for the introduction, given lifestyle-related healthy issues and chemical drug side effects, natural bioactive substances are valued. Ginger's rhizome is widely used, but its leaves and flowers are underexplored. Phenolics and flavonoids are key antioxidants, with extraction effect by solvent and plant part. And lack of systemic research on their antioxidant activity and sex general quantification exists. So using HPLC, this study compares extracts of ginger rhizomes, leaves, and flowers, screens optimal extraction, and quantifies 6 ginger to support comprehensive ginger utilization and functional food development. And for the research objectives, we have 3 objectives. So the first one is compare the effects of different solvents on the content of photochemicals and in virtual antioxidant activity in ginger rhizomes, leaves, and flowers. The second one is screen the optimal extraction solvent and quantitatively analyze the content of 6 general in different ginger parts in via HPLC. And the last one is verify the application potential of ginger leaves and flowers as antioxidant raw materials, providing experimental data support for the development of ginger-based functional foods or natural antioxidants.
Now I will move on to the second part of the poster, which focuses on the research design and methodology of this study. This research used a two-factor experimental design to evaluate the effects of solvent type and ginger plant parts on extraction efficiency. 3 solvents were compared, including 50% methanol, 50% ethanol, and water. And these were applied to 3 ginger parts: rhizome, leaf, and flower. As you can see from this graph, rhizome, leaf, and flower. The main objective of this design was to determine the optimal extraction condition and to identify which ginger part contains the highest level of bioactive compounds. In terms of analysis, the study focused on 3 key parameters: phytochemical content, antioxidant activity, and quantitative determination of 6-gingerol, which is the major bioactive component in ginger. For sample preparation, white ginger was used, and each ginger part was prepared in 3 parallel samples. All samples were dried and purified within 4 hours after collection to minimize chemical degradation. Regarding analytical techniques, UV-vis spectrophotometry was used to measure total phenolics and flavonoids, while antioxidant activity was assessed using DPPH, FIP, and PTS assays. Finally, HPLC with a C18 group was employed to quantitatively analyze 6-gingerol using a standard calibration curve. This methodology allows a clear comparison between solvents and plant parts, and demonstrates how spectroscopic and chromatographic techniques are applied in real analytical chemistry studies.
Okay, let's delve into the discussion and report of our poster. Start with the top left, antioxidant results. Ginger leaves part with 15% ethanol perform best. Tied to the phenolic and flavonoid compounds that scavenge free radicals. and this aligns with the spectral photometry principles. Leaves naturally accumulate this bioactive component to defend against the environmental stresses during the photosynthesis. It also highlights underutilized ginger leaves as a promising natural antioxidant source, with a potential application in function food or natural cosmetics. And let's move into the HPLC data below. It challenges the common misconception that raisins are richest in gingerase. Actually, leaves actually contains far higher levels. Our HPLC method, tailored to ginger's properties, follows chromatography separation theories, ensuring reliability. And this is partially driven by the light expression of key enzymes in gingerose biosynthesis. This finding paves the way for high-value use of the ginger's byproducts, such as developing nutraceuticals or food additives, linking lab results to real-world application. In summary, this result redefine ginger's potential, bridging analytical chemistry theory with a resource optimization.
Hello everyone, my name is Chen Ziyu. Today I'm excited to share a surprising discovery about ginger. My part is conclusion and the application prospect. The bottom. Ginger, one of our most famous species. We've always focused on ginger reasons, but this research reveals that ginger leaves, often overlooked, but actually a powerhouse of antioxidant. Through experiments using 50% ethanol, 50% methanol, and water as solvent, we found ethanol extract delivered the highest levels of bioactive compounds. Ginger leaves outperform both flowers and raisins across all key metrics. It means that ethanol's purity is better suited for dissolving active compounds in ginger. It changes the rule. Ginger leaves are not just a waste. They are a neutral, potent oxidant source. This discovery confirms that ginger leaves have overlooked high value and can be used as a neutral oxidant in the development of the function foods. In short, this study unlocked ginger's untapped potential. In the future, extraction process can be further optimized to promote the comprehensive and efficient utilization of ginger resource. My part is finished. Thank you.